Hello, and welcome to our presentation of organelle mutations in Arabidopsis thaliana. My name is Renee St. Jakes, and the assistant professor I worked with is Dr. Maolon Wang, and we're from the biology department at Westfield State University. So for this experiment, we decided to focus on mutations because mutations are the main source of genetic diversity in several types of organisms. We decided to mainly focus on indel, insertion deletion mutations, and point mutations because they're not often studied because how difficult they are to identify with the technology we had up until now. We chose to work with Arabidopsis thaliana because it is a model organism for most plantain studies and has a high rate of spontaneous genetic mutation. So we will be focusing on the indel and point mutations in this plant in this study. Decided to look at the organelles, chloroplasts, and mitochondria because they're not often studied in most plants because once again they are hard to identify. The chloroplast genome has a lot more copies per cell than the mitochondrial genome, which makes the chloroplast a little bit easier to identify and to work with. But overall, both of them are not very studied often because of how hard they are to see. So this is just a quick little flow chart I made of the process that we went through in preparing the data and then analyzing it. So to start, we would prep the data, we would check the quality of the DNA, we would add barcodes to it, we ran it through gel electrophoresis, added the barcodes to it to run it through the MinION. We ran the MinION for 24 hours, and then we began the process of analysis. So for our materials and methods, we used the MinION to run our analyses. We used the gel electrophoresis machine and the one-drop machine to check the quality of the DNA before we added the barcodes. Then we ran our data analysis mostly through Cyverse, IGV, and RStudio. However, we did need to use text editors such as OneNote, or we used Excel for some portions of the analysis, just because sometimes the files, once they were created, needed to be edited to be more workable with the software that we were using. So we checked the quality of the data of the DNA. We added the barcodes. Adding barcodes allows us to do one group sample instead of having to do each gene or strand of DNA individually. We can just pool them all together and run them all at once through the MinION instead of having to run them all at different times. We can run them all at the same time. So we ran the MinION for 24 hours. Then we began the analysis once we received our data back. So we had to convert all the files collected from the MinION to FASTA files and then edit them in text editors because there are some weird additions that were put in, changing it to a FASTA file or what have you. Any imperfections, we took them out. Once we did that, we used Cyverse and we used the BLAST feature on Cyverse, listed underneath the BLAST programs used on Cyverse section of this slide. We used create BLAST database. We created databases for all of the different barcodes with the different strands of DNA attached to them. Then we used BLASTDB CMB batch, which allows us to pool all of the data together, and then Minimap, which maps all the data. Once that was finished, we put it all in PowerShell to remove duplicates. We edited the files one more time in RStudio, just to remove any imperfections. We downloaded the GenBanks of the example DNA we had collected to create the databases in the beginning steps of this process, and then we used the IGV program to compare the data we collected to the GenBanks of the example data. So here's a picture of our results. On the left, we have chloroplasts, and on the right, we have mitochondria. As you can see, as I previously mentioned, the copies per cell makes the chloroplasts appear to have a lot more data in it than the mitochondria. The chloroplast is bigger than the mitochondria. To get further into the explanation of what the program is showing us, so along the bottom, we have where it says sequence, there's this strand of letters. That is all the nucleotides. We have A is green, C is blue, T is red, and G is this orangey yellow color. And each rectangle is a chunk of DNA. So the size of the rectangle represents how much information was in each rectangle. So they can go from 0 to 10. If you can see in bar 3, around the 78,680 BP mark, there is a very, very small rectangle. Oops, let me flip back. There's a very, very small rectangle. And as opposed to around 78,700 BP in that same row, that has a very large rectangle. That means there's more information in that rectangle. And some of the rectangles are split up and have multiple colors in them. This is because if you bring up this text box, were different nucleotides found in each rectangle. For example, in the bar 3 section, around the 78,700 BP mark, the rectangle with all the blue in the middle of it, there was one A, 4 C's, and 1 T. And if you go all the way down to the bottom where the example sequence is, you can see that there should be a C there according to the example sequence. So there is evidence that it has mutated slightly. This is just the mitochondria, as you can see. Very short rectangles, very infrequent rectangles, a lot less data is in there. But it's the same concept in analysis tools as the chloroplast example. Once again, here's the little box that tells how much of each nucleotide is in each rectangle. So to further analyze our data, we decided to move everything into a VCF file, which shows all of the different barcodes that we collected compared to each other. Along the left side, it will tell you if it's a transition or a transversion mutation. So in row 33, all the way to the left of the image, you'll see that it is an A is the reference and G is the alternative, meaning it's a transition mutation. And then in row 37, it goes from T to a G and a C, meaning that'd be both a transition and a transversion mutation. On the bottom, we have highlighted the homozygous mutations that we have found. Those are highlighted there 1-to-1, and the rest and everything else you're seeing that's not highlighted is a heterozygous mutation. And this is the chloroplast. And this is the mutation of the mitochondria. As you can see, I know it's kind of squished because I had to move some stuff around, but same thing. G to T, there are a lot of A to T, there are a lot of transversion mutations in the mitochondria, and once again, they are all heterozygous. Moving forward, and so we did find evidence of indel and point mutations in our data versus the example data. We were able to pinpoint where the mutations are in the genome using the IGV as an example, with using our example data as a gen bank. We were able to use the IGV to compare the two, and we were able to confirm the usability of the nanopore for undergraduate research. I found it to be a very easy process. We did have some problems with the analysis portion, but that's because my professor was on a Mac and I am on a Windows, so the two programs were clashing with each other. So I would definitely recommend completing all your analysis on one type of system as opposed to trying to use different ones, because it doesn't translate very well. But we were able to confirm that the Nanopore MinION would be good for undergraduate research. And a further experiment that I would propose based on the work that we've done is taking a particular gene or trait, pinpointing it, and then using the MinION to compare that gene or trait in different individuals, or using different individuals versus a baseline example gene. And here are my citations. I would just like to acknowledge and thank Dr. Maolong Wang for helping me with this experiment and for allowing me to conduct research with him. And I would also like to thank Tri Beta Honor Society for letting us work with their honor society and the support from everybody in the honor society. Thank you.